Inside the nucleus.
Inside the nucleus. Okay, good. All right, referring back to Model 1, what polymer is synthesized during this process? Messenger RNA. Now, technically, this is a pre-messenger RNA transcript. It has not been finalized yet, okay? So pre-messenger RNA. What monomers are used to build said polymer?
Adenine, thymine, cytosine, guanine.
Which were called what? Nucleotides.
Okay.
Remember from organic chemistry worksheet, those are the nucleotides. So that's what's being used to build that. All right, number 3, according to Model 1, what enzyme is required for this?
RNA polymerase.
If you're making a polymer, it's a polymerase. Polymerase. ASE means enzyme. All right, an RNA polymerase. Number 4, referring back to Model 1, what is the base pair rule for messenger RNA? What nucleotides pair together?
C and G and A and U, or A and T.
Right. So if the DNA has an adenine there, the corresponding RNA would be
Uracil.
uracil, U, not thymine, okay, because this is looking at RNA. So the normal DNA coding, because DNA is always double-stranded, is adenine binds with thymine and guanine binds with cytosine. But if we're looking at a messenger RNA transcript, adenine will bind with uracil. Uracil replaces thymine for that process. Guanine still binds with cytosine. No differences there. Now, DNA does not have uracil. Does not recognize that. So that's one way in which enzymes can tell the difference. Okay? Okay, so it says compare this base rule with that of 2 DNA strands. DNA does not have uracil. Which strand of the DNA contains the blueprint from that model?
The third end, the 3 end.
5 prime to 3 prime.
3 prime. Yeah. 3 prime.
Wait, we have 6?
We're looking at number 5.
Oh, 5.
If you look at number 5, then they have some names on there, and the name here says template. That's the reading strand. That's the reading strand. So that's the one they're looking for there. Now in number 6 then, it says in which direction is the DNA model read?
Oh, 3 prime to 5
3 prime to 5 prime. What is 3 prime to 5 prime? What is that referring to? I've been trying to get across to you all semester by making you do this on the structures that we draw. Whenever I made you draw glucose, what did I make you do? Labeling what? Carbons. I had you count the carbons around there, right? So we started with carbon 1, we ended up with carbon 6. When I had you draw ATP, you also had to label the carbons. Right? So when you come around there, you had 5 carbons. So this is what we're referring to is where does a phosphate group get added to that ribose? It can be added to carbon 5, or it can be added to carbon 3. 5 prime is referring to the 5 carbon. 3 prime is referring to the 3rd carbon. So that's what 5 prime and 3 prime are referring to. Is like, where is that being added? Where is that strand growing with that polymerase? So I'm hoping you're starting to put this stuff together. We've been studying all semester, and now you can actually use that to go into these other areas of intense study, such as transcription and translation. Okay, so 3 prime to 5 prime, that's how it's being read. 6B, the DNA strand and pre-messenger RNA are anti-parallel. They're running in opposite directions, but they are parallel, right? With this in mind, label the 3 prime and 5 prime ends of that strand in your model. All right, were you able to find those?
Mm-hmm.
Okay, they should line up in opposite directions. In which direction is the pre-messenger RNA molecule constructed?
5 prime to 3 prime.
5 prime to 3 prime, yes. Okay. Number 7. Before printing presses were available, books had to be transcribed in order to share the information. Considering the definition of transcription, explain what the process in Model 1 is described using that word. To just transcribe is to make an exact copy. To make an exact copy.
Oh, sorry.
So that's what we're doing.
I don't even
Yes.
I just signed
And we're trying to make an exact copy, so each one of those has to be exactly. All right, in the next section, it said in eukaryotes, the enzyme RNA polymerase joins with several transcription factors. We've been alluding to transcription factors. These are things that actually attach to the DNA to say we're supposed to read here. We're supposed to read here. And so, well, we'll get to those in a minute here. So we've been kind of talking about them. I go into more detail in cell biology with those. They're transcript factor proteins, right? And they hang out at what we call the promoter region on the DNA. A promoter region attracts in all the transcription factors for those genes that fall behind it, the codings for genes, because we're actually looking at this process where we go from the DNA to a copy of the DNA to eventually a protein product. That's the goal. We want a protein product that's going to do a job inside the cell. Now, we've been talking about that protein could be a receptor on the outside the cell. That protein could be a portion of the MAP kinase cascade. That protein could be an enzyme that's going to help us make a GLUT transporter to bring in sugar or to break it down, okay? So that's the product. That's what we're trying to make. So there's a segment on this DNA that codes for that. And so this promoter helps these transcription factors come in, and we call that initiation complex. Okay, number 8. What parts make up the transcription initiation complex? What would be the enzyme that we would need?
RNA polymerase.
RNA polymerase. What else do we need?
The proteins.
Transcription factors, right? They're coming in. And they're the proteins. They're the proteins. And where does it complex form?
In the nucleus.
Inside the nucleus, on the DNA. What segment of the DNA? Promoter.
Yeah, I said that to everybody would.
Okay. Nearly all cells in an organism contain identical DNA. Everyone. Each DNA strand may contain hundreds or thousands of genes. It is likely that a cell would transcribe all the genes within its nucleus simultaneously. Justify your answer. Do we copy all of them simultaneously?
No.
Why not?
So then there won't be like a master copy left. Like if we
It
like.
says it only copies it when it's needed.
Okay, so if you need a protein to bring in, to make an enzyme to bring in glucose, you're only going to make that one. If you need a receptor, you're only going to make that one. If you have a MAP kinase activity that has been stimulated by some sort of hormone or even insulin, you're going to make what it tells you to make. You'll have certain transcription factors that will go into the nucleus and adhere to the promoters to promote that protein production. Okay, so it's only going to make what you need. Now on number 11 then, it says, considering the many types of cells, you're a multicellular organism. The Raj has brain cells, neurons are firing. You don't have many fat cells. I got the fat cells. We got muscle cells. We have liver cells. Each one of those organs has a particular composition of cellular activity as well as tissue layers. Each one of those, based on their differentiation, have a job to do. They don't need to express all their genes all the time. But each one of those cells has the blueprint for the entire body. And if we can find a way, that's the whole aspect of stem cell technology. Can we take a cell back to its stem cell state, an undifferentiated state, and have that create new liver cells, or have it create new neurons? Have people who are paralyzed walk again because we can create new neurons. That's where the research is going right now. We're trying to figure out how to reverse that process. But right now, in a multicellular organism, those tissue layers, those organs, and those cells are devoted to one job. So they don't need to express every gene in the body, but they do have them. So on number 11, it says, considering the many types of cells in these multicellular organisms and their different functions, is it likely that all cells transcribe all their genes at some point?
Yeah. Possibly.
But if you're a muscle cell, you will never be a neuron. You will never transcribe those genes. You have them. But during the lifetime of a muscle cell, it won't do those other jobs. So we're going to say certain genes will be switched on in specific cells. And then others, depending on the location and function of the cell. So while some cells may use all their genes, other ones will never. They just won't do it. Neurons, like we said, they basically stop at the G0 stage in the cell cycle. They will not go further. And so they will never create actin or myosin or a sarcomere. Okay, they will never use those genes. So like I said, we do have some cells that are more general purpose, maybe epithelial cells, but other cells are highly, highly differentiated. Okay, now we said we had to get to 15 here. Number 12 says compare the pre-mRNA to the mRNA leaving the nucleus. This is what I wanted to bring this up.
Hey, Logan.
Hey, what?
Go through the screen too.
Oh, yeah. I did want to do a little, hold on one second. I want to go back a little bit.
Hey, Jess.
Remember, all of these, we're on the PowerPoint. You can use the PowerPoint to find all of this information.
Can you hold this for me for 2 seconds? Thank you.
This is just a reminder, when we covered the polymerase, there are 3 different polymerases for interaction. You guys don't have to know that, just recognize it's RNA polymerase that does the job. But there's a lot of different types of transcription factors. These are the proteins that help bind at the promoter region to have that initiation complex. So basal factors, these are the ones always at the promoter. We have those that are upstream from that site and those that are downstream from this site, and then we have those that are inducible. And so inducible basically says they're time or tissue specific, right? They can be induced. It's like, why make an enzyme to break down lactose, right, if you never drink milk? So it's inducible. We'll see this with bacteria. If they're exposed to lactose, the lactose will actually go in and bind to some of these factors. It's an inducible factor, and they'll start producing the enzymes to break down that sugar. So here's what it looks like. You'll see this in the book on, I think, can't remember, it's when you get into the genetics chapter. Okay? Here is a core promoter region. There is an area up here called the Tata box, right? This is something that's very unique, and it's usually a certain distance from the actual protein coding region, but it's pretty much the same in all organisms. In other words, it has thymine, adenine, thymine, adenine, thymine, adenine, thymine, adenine, right? And it's recognized by several of these binding proteins. Here's our RNA polymerase, right, in the core promoter region. But the actual RNA polymerase is one of the last things to bind here. So just for your information, here are all the transcription factors that come into play attaching to this Tata binding region, okay? You'll notice there's some other activators that attach here that'll actually bind other segments upstream. So this is upstream of that box, so there's enhancer regions, and it'll enhance the copying of this segment, whatever this gene is that we need. The RNA polymerase then is brought in by these guys right here. These are basal transcription factors that are binding here, and they bring in the RNA polymerase. Once the RNA polymerase is there, it's time to get started. And one of the last guys here, this guy here, H, he's going to basically phosphorylate RNA polymerase and get it going. Remember, phosphorylation turns things on, it activates things. And so once that happens, all of this falls away, and the RNA polymerase starts chugging down here, copying that DNA into RNA. Okay, now there's the coding region downstream from that side. So I wanted you to see kind of a visual of that. We've been talking about it in the worksheet. Okay, so this is more of a simplistic Here is the upstream region, so the top box would be here. Here's the promoter region, and here's downstream where the RNA will be copied into. So again, you can find all of this on the PowerPoint to help you understand that process. Now, this is for the new material here. When you're making your pre-messenger RNA, you will have segments that aren't needed for the final product. So here's our upstream regulatory elements. They may be enhancers, they may be silencers, etc. It just depends on what's binding up there. Here's kind of our simulated promoter region. So that's where the RNA polymerase is going to start at. Okay? And this is the coding regions. Now, you'll notice here that we have, there's our start site, exon, intron, exon, intron, exon, termination sequence. Not every one of these segments is a gene that's coding. What gets cut out?
The introns.
Introns. So they're in between the exons, which will exit the nucleus. So it's going to cut out these introns and put the exons together, and that will exit. That is now preparing our true messenger RNA. That's the true message. So these other segments have other jobs to do. We're not going to talk about them here. Some of them have just been discovered. But it shows our coding regions there. And that is called a transcription unit. Go down there. So here's messenger RNA processing. So up above here, they show the exons in purple, the introns in white. So a gene is composed of exons and introns, and it's been transcribed. But then processing involves these ribozymes, these little proteins, that will actually move the introns from this segment, splicing these exons together, bringing those together, and then now we have a messenger RNA transcript that can be read. It's going to do one more thing to it, and that's before it can get out into the nucleus, or out of the nucleus into the cytoplasm. There are different types of enzymes called nucleases. They're going to chew this sucker up, and that's their job. They chew up old messenger RNA strands because we don't need them anymore. Chews them up, and we recycle those nucleotides back to the nucleus. But we don't want this chewed up yet, do we? So we have to regulate it. We're going to cap one end. We're going to put a guanine cap on backwards on one end, and on the other end, we're going to put a whole bunch of adenines at the end. Adenine, 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 adenine. All the way, thousands of adenines. And that way, that nuclease is busy chewing up the adenines at the tail while the rest of it's getting copied. Okay, so that protects it. So once we get the exons together, then we're going to cap it and tail it for processing. So here's, look at this. Removal of the introns. It's called a spliceosome that does that job. Oh, and they're all together. Now you'll notice there's a guanine cap here on the end and an adenine tail. And now this is truly messenger RNA. Did you like that, Anna? Did you think my animation? Yeah, you can tell I've never had a graphic design class. There we go. And there it is together. Oh, that's fabulous. And so here again, another schematic of this. So it talks about having a 5' cap. So on the 5' end, we're going to cap it with a guanine. And on the 3' end, we're going to put a polyadenylated tail. Just add a bunch of adenines on there. We take out the introns, we splice those out, we bring the exons together, and now we have a piece of messenger RNA that's mature that can go out into the cytoplasm and be read by what? What's going to read it? What's going to make proteins?
Ribosomes.
Ribosomes. And the ribosomes can be free, or they can be attached to our endoplasmic reticulum. So what's the function of a polyadenylated tail? Protect it from who?
You.
What's the enzyme that chews up nucleotides? Nucleases. All right. So now let's answer these questions. 12A. What has been removed from the pre-messenger RNA? Introns. B. What has been added to the messenger RNA that was not present in the pre-messenger RNA? Yes. And how will it leave the nucleus? The pore. Okay, and that pore is going to regulate what can come in and out. Okay, 14. The first nucleotides on the messenger RNA will be read in the next step to produce a polypeptide. What is a sequence of bases? AUG is always the start sequence. Adenine, uracil, guanine. It will code for methionine, and an amino acid, which we said, remember learning your amino acids, it's a special amino acid, it's a start codon. And 15, the M in messenger RNA stands for messenger. Why is this molecule called the messenger RNA? And it takes it to the cytoplasm for translation. Okay.
What happens to the introns?
They are recycled. Broke down and recycled. It's one of those things you don't often think of.
Like broke down all the way, or they go back into the pre-RNA?
No, they're broke down all the way. Yeah, the enzyme just chews them up. All right. I think we got everything that we needed there.
Wait, what is the point of that?
They actually have some regulatory sequences on there. So while we may not have more genes or DNA as other species, we have a very unique way in which we can combine those together based on inducible factors that would attach to those. So it may only like copy certain segments on there, or it may bring them together in unique ways. And that's how we can make a very diverse form of proteins for different metabolic functions. Okay. Transcription's done. What now?
Translation.
It's time for translation. We are now in the nucleus here.
But are we doing the back half of this worksheet or getting the other one out?
We're going to get the other one out. Translation.
Has Erica graded anything?
Hey, Professor, do you want us to show you the evaluation they give their class?
What's that?
The evaluation of the course.
Oh, yeah. After. Okay. The second stage of this protein production is called translation because we're translating that mRNA script into a protein. All right, so this is a very simplistic version of this process. But the messenger RNA will engage with ribosomal subunits, all right? And so we often see ribosomes as being together, right, coming in together, but they're actually not. The subunits are actually separate, and they're brought in to the messenger RNA. And we have, just like we have initiation complexes on the DNA, we have complexes on the messenger RNA that'll bring in the ribosomes and tell them, copy here, okay? So here's our AUG segment. You'll notice in front of that is a 5' region, so we know the guanine cap is over there, and out here somewhere is our polyadenylated tail on the 3' region. So the AUG segment's going to basically bring in a methionine amino acid. It's attached, though, to this structure. What is this structure called? Looks like a T. And it transfers in the amino acid. tRNA. Okay, so this is another type of RNA that's coded for in the nucleolus region of the nucleus. Now, when the tRNA, the transfer RNA, gets into the cytoplasm, it'll get charged with an amino acid based on its anticodon region. So it's going to have methionine, and then that will line up with AUG. So methionine, we said, is our start codon. All right. I can't believe it. Is this maybe? It's one of my very first lovely pictures. So here we have our subunits of our ribosome. These small subunits can be a 30S or 40S subunit, depending on if you are bacterial or if you are eukaryotic. Okay? Same with the large subunit there. But they are not additive. Now, the S stands for Svedberg unit. If you're going into chemistry or biology, you'll start getting used to that. It's a sedimentation coefficient rate. So if you put things into a centrifuge, they will sediment out based on their size and their shape and how bulky they are, how dense they are. Okay, so this is not a unit that you're probably familiar with, but it basically has to do with centrifugation. So if it's very bulky, right, and doesn't have a lot of weight to it. It may not centrifuge down very far. But these two units will actually bind on the messenger RNA. But there are two complexes. On there, you have 3 different regions on the ribosome in which tRNA can come in. So we have the E site, which stands technically for empty. We have the area in which we're making a peptide bond. And then we have the A site where things are arriving into. And there's our methionine right there at the AUG codon. All right, so the tRNA brings the amino acid into the ribosome. And there's kind of our tRNA. You'll notice the tRNA is made of nucleotides, just like messenger RNA, and it folds back on itself into this T-like structure. Okay? So this is a strand of RNA that's been folded back, and so it's going to have the attraction in these certain areas where it basically comes together. See the bonds? Where we have A binding with U and guanine binding with cytosine. Down here is the region that's going to line up with the codon, the code on the messenger RNA. So this is called the anticodon. So again, it would have the same sort of binding. If the codon starts with A, the anticodon would start with what?
T. U.
U. There we go. So that's how we're going to set up the anticodon. So that complements the messenger RNA. So it's usually a 4-lobed structure. It's got the same orientation. It's got a 5' end, and it's got a 3' end. The amino acid is charged to the 3' end. The lobe down here, the bottom lobe, is where the anticodon will be established. And it is complementary to the codon that will bind it. So again, you got to get used to that 5', 3' structure. Here is more of a detailed loop, and they show the different loops. Here's the D loop and the anticodon loop and a little variable loop there. And this is the region up here at 3', that's the acceptor region for the amino acid. And so the amino acid would get charged or added to that segment. The amino acids have to be in a correct order. What order did we call that in chapter 3?
Wait, what order? What?
When we talked about making a protein, we said there's a certain sequence of amino acids. What level did we call that?
Secondary?
Primary. That's just a sequence of the amino acids. Now, secondary talks about some of the folding, an alpha helices or beta pleated sheet. And then when it gets more complicated, we have more folding back, it's tertiary, okay, the 3rd level. And that's when it can become active or even at the domain level. So, these tRNAs line up with amino acids using that messenger RNA code. Reading the code, every 3 bases will make what we call a codon. So we say it's a coding triplet on the messenger RNA. So every 3 bases will be read as a codon, and that will code for a specific amino acid. Okay? If we have a 3-letter code, and we have 4 different bases, adenine, uracil, guanine, and cytosine, that gives us 64 different codons that could exist. We also have 3 codes for stops. It says stop here. Stop transcribing or translating. And so those would be nonsense codons. So that gives us 61 codes for amino acids. All the different amino acids. How many amino acids did we have that we learned? 20. 20. All right. So 1 amino acid codes for initiation. That was methionine. That was one of the special ones that you learned. We have 20 amino acids. So we have more than 1 3-letter code per amino acid. And that brings up our codon table. Don't have to worry about that. You guys learned all those already. Here's the codon table. You have one of these in your book, and I believe they gave you one in your worksheet, right? Yep. Front page. This is the codon table. So if you like secret codes, this is for you. Now, to read the code, we're going to start off with AUG, since we know that's the start codon. We're going to start with the first position over here on the left-hand side. And here are all the codes. We have guanine, adenine, cytosine, uracil. So it goes, the 2 purines are down here, the 2 pyrimidines are up there. So our first one was A, right? So we know we're in this segment right in here. A. The next one was U. If we go across the top, that's where the second position would be identified. U, C, A, and G. We know the second one's U. So far we're right in his section right here, right? A, U. The 2 possible amino acids are isoleucine or methionine. Notice methionine has a little asterisk next to it. And so now let's come over here to our third position, G. So they have all these repeating in each one of these different columns, okay? So on our AU segment, if we look over here, the third one's G. That must code for methionine. Okay? Fourth one, methionine. And that one we see is special. That's our start codon. And start. Okay? Now if we're looking at another one, let's say the U, A, G codes for a stop codon, a nonsense codon. In other words, it does not code for an amino acid. If we look at, let's give an example. Lysine's always good. Where's my lysine? Or leucine. Look at leucine over there. Do we see any more leucines over here? We have 2 different columns there of leucines. Leucine could be UUA, UUG, CUU, CUC, CUA, or CUG. Why would we have so many codons for this particular amino acid?
So we make a lot.
Make it a lot, maybe?
So we make sure we make one.
What's that? Maybe we have a lot of it? What if you had a mistake? These enzymes might make mistakes. Leucine might be a very important amino acid. And then you have a high percentage rate of being correct. Now, you'll notice there's only 2 possible first positions you can have, C or U. It only has 1 possible second position, that's U. But the third position, high variety. You could have any of them in the third position and still get leucine. Where do you think the most incorrect activity occurs? On that third position. It doesn't fit quite right? When the enzyme's coming in there and there's a little bit of wiggle room on that third position. And so this ensures that we will get leucine every time. Doesn't matter. As long as we have any of those, we'll go through there. Arginine, same thing. Okay? You could have a C or an A for the first position. The second position has to be a G, but you could have pretty much anything in the third position and still get arginine. All right, so that's kind of here's the wobble concept right here. Where it looks at this one, okay? What the heck is that? You're doing what? And then the fence. Well, that's a good explanation. What is that sound? All right, so here's where our flexible pairing is, right here on that third position. Okay, so now I want you to work together next 5 minutes and make progress through, where's my glasses here? Progress through number 6. Number 6.
Already got it.
Good job.